So now in section 4.2, we're going to give an overview of cell structure. When you talk about living things, we can really characterize or categorize them into two basic groups based on their cell structure. You have prokaryotes, which are simple cells, although not doesn't mean they're not complex, but based on their level of organization, prokaryotic cells are much more simple in nature than eukaryotic cells. And so you have living things that consist of one prokaryotic cell, like bacteria and archaea, and then you have eukaryotes, organisms that are eukaryotic in nature, which can be single-celled, like a protist or a yeast, or they can be multicellular, like plants, like animals, etc. So in this chapter, we're going to compare and contrast prokaryotes to eukaryotes. Now, prokaryotic cells, as we said, are simple in nature. By definition, they lack a membrane-enclosed nucleus. Pre-nucleus is essentially what prokaryotic means. It doesn't mean they lack genetic material. Bacteria have genetic material just like all living things do. But how that genetic material is housed within the cell is very different. There are two categories of living things that are prokaryotic in nature. There's bacteria and archaea. Both are small. When you compare them, what I would say about bacteria is they're more ubiquitous. We find them in our daily lives. They're in the yogurt that you drink. They're covering your skin, in your colon. There are more bacterial cells on and in you than you have human cells. There's a human microbiome, which they're trying to elucidate, which means that they're trying to figure out who are all these organisms that live on us and thrive on us and benefit us, which is what we often don't think about because most are not harmful. Archaea are in niche areas, tend to be living. They tend to live in extreme environments, high heat, high salinity, high poisonous gases, where most living things would expire due to an interruption in metabolism or an effect on proteins, etc. pH, extreme in pH. Archaea can inhabit those environments. When you look at a typical bacterial cell, we'll use that as our prokaryotic example. They have a simple plasma membrane which delineates the inside or separates the inside from the outside environment. It's a barrier, so while some things are rejected, other things get through, and vice versa. We also have a fluid interior called a cytoplasm, in which all the important structures of a bacterial cell are housed. We also have a nucleoid region. That's where the DNA is found in the cytoplasm. And then we have ribosomes, which are involved in protein synthesis. So a typical bacterial cell is a lot like a studio apartment. Now, you may not be familiar, but a studio apartment is a one-room apartment. So your bed is also your couch. Your living room is also your bedroom. I mean, it's just literally one space which you reside. That's what a bacterial cell is. All the important structures, all the important functions occur within that cytoplasm. Now, there are structures outside the plasma membrane that make bacteria unique, specifically their cell wall, which is made of peptidoglycan, which is a sugar protein moiety, meaning it's a mixture of sugar and protein. It's that cell wall that provides support and protection from the effects of water on the cell. So a cell that lacks a cell wall, as water rushes in, the cell gets larger and larger, and at some critical point, the cell bursts open. It lyses. But now if I put a cell wall around it, the cell can only expand so much before the cell wall prevents further expansion, thus protecting the cell from bursting. We also have an additional layer called a glycocalyx. It prevents a bacterial cell from drying out. It aids in protection from host destruction. Any Harry Potter fans out there, it sort of acts like an invisibility cloak, so the host's immune system can't recognize the bacteria. It also can aid in attachment, so it allows a cell to attach to a surface. And then there's other appendages, protein appendages. There's pili, 1L, which aids in attachment, flagella, which aids in motion. So here are those important structures. I think on any exam, if I give you a bacterial cell, you should be able to label all its parts and tell me exactly what those parts do. Now, I think my picture is, I mean, look how fancy this is, but if you're into these more, elaborate pictures, here certainly outlines all these important structures. Eukaryotic cells, more complex, certain, larger, contain a nucleus which houses the DNA. So eukaryotic cells were named after the fact that they do have a nucleus. We're going to see the cell is compartmentalized by membrane-bound structures called organelles. And each organelle has a unique structure and function. So remember I called the prokaryotic cell a studio apartment? So you slept in the same room that you entertained, in the same room that you studied, in the same room that you ate. I mean, every function of that apartment occurred in that one room. But now if I have a 3-bedroom apartment with a living room and a dining room and an eating kitchen, now I'm splitting up the functionality of the apartment by walls, right? I sleep in one room, I entertain in a different room, I study in a separate room, and then I eat in a room different from those. So when you look at the inside of a eukaryotic cell, it's separated by membranes, similar to the walls of an apartment. Now, while eukaryotic cells are larger than their prokaryotic counterparts, there's no one shape, there's no one size, there's no one level of organization that's common amongst all eukaryotes. They differ among species and even cell types of the same species, and you know that. You learn about the different cell types in anatomy and physiology, so I think you're well aware that's not all cells look and behave in the same way. You should know all these structures by now. I'm not even going to walk through each of these. I think it would insult you. For this chapter, you need to be able to label a cell. You need to be able to identify the different parts and tell me what each of those different parts do within the eukaryotic cell. Here's that great example. Cells can differ even though these are both human eukaryotic cells. We know a human skin cell looks much different than a human nerve cell. Most of the reasons being that the functions are very different. Skin needs to aid in protection, so the packing of those cells are very different compared to a nerve cell, which has long branches, which needs to transmit information in the form of electrical signals. Here's a plant cell. One of the other things I need you to be able to do is to be able to distinguish between a plant cell and an animal cell. While most of the organelles are common, we do have the presence of a central vacuole here in a plant cell. We do have the presence of a cell wall, which is very different than an animal cell. And we also have the presence of the photosynthetic machinery called a chloroplast. So you should be able, for a plant cell, identify all the structures, compare and contrast to an animal. Now, what determines the characteristics of these cells? Well, I'm going to introduce a concept that is threaded throughout the textbook, and that's the idea of genomes and proteomes. Genome represents the entirety of your genetic information. So all the genetic information that's carried within a cell is that cell's genome. The proteome represents all the proteins that are produced as a result of that genome. One of the things I'm going to tell you is that the skin cell and the nerve cell have the exact same genome, which you inherited back in a reproductive event at some point in your life cycle. But yet, man, these look so different. They behave so differently. But the information is the same. So how can two cells with the exact same information look and behave so differently? The difference is in how is that information read. How does that information produce protein? And the types of proteins and the amount of proteins produced by each of these cells makes them very different from one another. And so that's going to be a common theme throughout this semester, is that the genome dictates the proteome, and the proteome makes a cell what it is, it makes you what it is. And so by regulating the expression of the information, by regulating the amount of protein, the sequence of that protein, how that protein is modified, can impact ultimately how those proteins function. And it's changes in the proteome as a result of mutations in the genome that causes, for example, cells to behave abnormally in the case of cancer. So I want you now to start to get used to this concept that while the information is critical, obviously, it's how that information is read and expressed that allows us to have different cell types within an individual organism.